Good day, everyone. Today, this video is talk about the proteins, the structure, properties, and biological significance of proteins. I'm Mr. Ong from College Tingkatan 6 Sediputra. Previous lessons, we have discussed about the lipids, the types of lipids, triglycerides, phospholipids, and also steroids. Then these videos will talk about the classification of amino acid into the four main classes. And we also describe the structure of an amino acid and the formation of the dipeptide, okay? And also the formation of polypeptide. And then we also discuss the properties of proteins such as amphoteric, isoelectric point, buffer, and colloid. And we also differentiate the various levels of organization such as primary structure, secondary structure, tertiary structure, and the quaternary structure. And we also classify the proteins according to the structure, composition, and functions. And at the end, we also explain how is the proteins denatured and renatured. Proteins are molecules. They are macromolecules, okay? They're composed of amino acid. So that means compared to the lipids, so proteins, they are macromolecules and also they are polymer. So the monomer of the proteins are amino acid. And then the proteins will always contain the elements nitrogen, nitrogen as well as the carbon, hydrogen, and oxygen. So sulfur is often present too because the amino acid cysteines, okay, they might found in proteins. Then a protein is a biologically functional molecule that consists of one or more polypeptides, and each folded and coiled into a specific three-dimensional structure. For example, hemoglobin. Hemoglobin is a functional molecule, okay, proteins which contain 4 polypeptides. The function is used to transport oxygen and carbon dioxide. And proteins also involved in all aspects of the metabolism, okay, especially the enzyme. Most enzymes that can accelerate or speed up different biochemical reactions that take place in an organisms or in a cells. Okay, so most enzymes are protein. Remember, not all is most enzymes are proteins. Protein also can be as the colloid, okay? So for example, plasma proteins. Plasma proteins, they are the large solute which can contribute to the osmotic pressure in blood. That means if the concentration of the plasma proteins in blood is higher, so they will have an osmotic effect, to draw water from the surrounding into the blood by osmosis. So this is called osmotic pressure. And the proteins are assembled into a variety of shapes, so allowing them into to serve as the major structural components of cells and tissue. And each cell type contains characteristic forms, distributions, and amount of protein that largely determine what the cells look like and how each function. For example, the proteins, most of the proteins they found in the muscle cells, they are myosins and actin. So these proteins are responsible for muscle contraction. So you will study this, the action of myosin and actin filaments in the next semester. So this is proteins hemoglobin. Hemoglobin. So they found in the red blood cell. So the hemoglobin is responsible for transport of oxygen and carbon dioxide. Amino acids are the subunit of proteins, or we can say the monomer of polypeptide. Amino acids have an amino group, okay, amino group here, and a carboxyl group, COOH, bounds to the same asymmetrical carbon atom, okay, carbon atom. So this carbon atom is called alpha carbon. So they have hydrogen atoms to bounce to it. So these three, an amino group, a hydrogen atom, and a carboxyl group. They bounce to the same asymmetrical carbon because the different group here. And also with a R group here, okay, or we call it as a side chain. So these four different partners okay, of an amino acids so like they are the amino group here, okay, the carboxyl group, and a hydrogen atom, okay, and a variables R group, okay, or the side chain we call it. So all the amino acid they have the similar or we call they have the common groups, okay, which are amino group, carboxyl group, and a hydrogen atom. So only the difference is at the R group or the side chain will be different. Amino acids are crystalline solid, so that means can be crystalline and dissolved in water, but they are insoluble in organic solvent. Okay, so for example, the amino acids that you can find in your kitchen, okay, the MSG, monosodium glutamate. So the glutamate is an amino acid, so it can be crystalline. So it's similar to that lah. Like the common one is Ajinomoto lah. Okay, so when the amino acid, okay, they are found in a cytosol, okay, in the cytosol of a cell, or we call cytoplasm. So the amino acids will exist normally in a dipolar ions form, or we call it as deuterion, okay, deuterion, all right. Deuterions means the amino acid, okay, they have a positive charge and also a negative charge group here, okay. So that means each carboxyl group donates a proton, okay, they donate a proton. Protons mean is hydrogen ions and becomes ionized as the COO negative. Whereas each amino group, okay, NH2, they accept the proton and becomes ionized positive charge. Okay. So, and because of these properties, okay, the amino acids are described as an amphoteric because they contain the acid and the basic properties. So this one is the acidic property, this is the basic property. Okay. So because the ability of the amino group, okay, and also the carboxyl group to accept and release protons, so the amino acids in solution, they resist the changes in acidity and alkalinity. Okay, so this one is very important because in our blood and also the tissue fluid, they contain large amount of amino acid. So this amino acid, they can act as the pH buffer. Okay, they act as the pH buffer to resist or to limit the change, okay, of the pH in the tissue fluid or blood. Okay. So the amino acids that tends to stabilize the pH as we mentioned just now. So actually how to do that? So in an acid solution, okay, acid solution, the low pH, so that means when the what we call the cytosol, okay, for example, the cytoplasm, okay, or the surrounding of the or in blood, they becomes acidic. Okay, so the amino acid they will receive, okay, because in an acidic solution, they have high concentration of hydrogen ions. Okay, so in order to resist or to maintain the change of the pH. So these excess hydrogen ions will be accepted by the carboxyl group. Okay, so it becomes COOH. Then the amino acids will become positively charged. So that means in an acid solution or in an acid solution, the amino acids tends to be positively charged. So in an alkaline solutions, then alkaline solution, we know they have a high concentration of hydroxyl group. Okay, so in order to reduce the concentration of the hydroxyl group, so the amino acid, okay, they will release, okay, or is donate, okay, donates the proton, okay, to the surrounding. So these hydrogen ions, they will react with the hydroxyl group, then to form water. Okay, so this the concentration of the hydroxyl group will be reduced. So this they can restrict or resist the change of the pH of a solution. So the amino acids are a very important biological pH buffer. And then this pH buffering, this pH buffering properties is retained even when the amino acids they are joined by the peptide bonds to form a polypeptide. This is because they have a free amino group, okay? All the polypeptide, they still have a free amino group and also a free carboxyl group, which are able to donate or receive the protons. Okay. So the pH at which the amino acid is electrically neutral. That means when a certain amino acid they found in a solution with a particular pH. So if the amino acid they show the electrically neutral, that means they are in a zwitterion form. Okay, in a zwitterion forms. So we can say that the pH is the isoelectric point for that amino acid. Okay. So regarding to this these properties, isoelectric points properties, we will discuss further in the subtopic of analytical techniques in biological molecules. Okay, we proceed to the physical and the chemical properties of an amino acid. So as you know, all the amino acids that have the same amino group and the same hydrogen atom and with a carboxyl group that join to the alpha carbon. So the amino acids they are differ in the R group. Therefore, the physical and the chemical properties of a particular amino acid, so normally will be determined by the R group or the side chain. So therefore, overall, they will affect the functional role in a polypeptide. So it is the R group and or the side chain. So there are many different types of amino acid. So we only study the 20 amino acids that found naturally, okay, to be used to synthesize proteins. Okay, so just now we have mentioned, okay, all the amino acid, okay, they have a different in the R group. Therefore, according to the R group, we can classify the amino acid, okay, into 4 main classes. Okay, the first one is non-polar. Okay, if the side chain is non-polar, so they will show the properties of amino acid is hydrophobic. If the side chain of the amino acids is the polar group, okay, they have a polar group, so the amino acid is hydrophilic. Okay, if they are charged, okay, they are charged, and then for the charge, they also can be either is a negative charge or positively charged. Okay, so the one is they show the negatively charged, so it is acidic lah. For example, COO negative. And most of that, they are positively charged, so they are basic. And then for others, they are charged, so they, together with the polar side chain, so the amino acids are not as hydrophilic amino acid. Okay. And then you must know that all amino acids have the carboxyl group and amino groups. So the terms acidic and basic in this context, we are referring to the side chain only. So for the polar example of polar non-polar amino acid, okay, the simplest amino acid is the glycine. Glycine is the simplest amino acid in which the R group has a hydrogen atom only. Okay, so they have no polar and also no charge. So that's why we call it as a non-polar amino acid. And then the other one is the alanine. Okay, that has a methyl group, which is a CH3, okay, as the R group. So the CH3, that means here they have also non-polar, okay, and also no charge. Okay, for example, can expand this one. Okay, you can expand this. So then here they becomes the hydrogen atom here. So balance distribution here evenly. So that means they cannot be a polar amino acid. Okay. So we can look at the other example for the non-polar amino acid. Okay. This one we already done just now, glycine, okay, alanine. And then look at valine, also the same. They have no polar or charge. Okay, same to here. So you see, all of these they have no polar and no charge. Okay. So for the polar amino acid, for example, serine, okay, that has a CH2OH group as its R group. So the OH here, okay, the OH here. So that means here is slightly is a polar, the polar. So that means that's slightly positive, here slightly negative. Because the oxygen is a more electronegative atom compared to hydrogen. So seems to the threonine, amino acid threonine. Okay, so this sulfhydryl. Okay, OH hydroxyl. Okay, this one they have the carbonyl group. Okay, and then also the NH. Okay, especially the carbonyl group. Okay, they are polar. So for the acidic amino acid, so two amino acids we have in one they are aspartic acid, okay, and the glutamic acid, okay, or sometimes we also call the aspartic acid if they are in charge form, we call it as aspartate. Okay, aspartate. This one is glutamate. We call it as glutamate. Okay. So they show the charge and negative charge on COOH. So this is acidic amino acid. So for the basic one, you already know like this one. Okay, the basic one, positive charge. Okay, lysine, for example. So this is a non-polar. Okay, see the non-polar amino acid. Okay, and also the polar. Okay, and charged amino acid. Okay. So when these amino acids these amino acids they are able to form a polypeptide. Okay. So first, the two amino acids they combine chemically. Okay. So by condensation, by the process called condensation. So to form a dipeptide. Okay. So the dipeptide actually is formed by two amino acid, and then they join by the peptide bond. Okay. So the peptide bond, this is the peptide bond, or we can write the peptide bond as like this. Okay, CO and then NH. So this is the peptide bond. Okay, don't forget the water molecule. Okay, because this is the condensation. And then from this reaction is reversible, so can be catalyzed by protease, okay, to hydrolyze this dipeptide into 2 amino acid. Okay, so the formation of this peptide bonds is from one carboxyl group, okay, of an amino acid with an amino group with another amino acid. Okay, so to form one water molecule. Okay, then this one like dipeptide, they form a dipeptide. So from here you can see clearly clearly. So how's the water is produced? Okay, a longer chain. Okay, let's say we add the amino acid again to the dipeptide, so they form tripeptide and so on. Okay, so at the end they will form a polypeptide. So the polypeptide actually is like this so they have one N terminus and the C terminus. Okay, and then you can see here actually is the polypeptide backbone. Okay, the backbone of polypeptide. So you can see the difference is here, the R group. Okay, the R group are different. Okay, so each polypeptide that has a free, like just I mentioned to you. Okay, N terminus, and the free carboxyl group with at the other ends is called the C terminus. Okay, and then from here you can see the terminal groups. Okay, and also the difference in the side chain. Okay, and then this the whole polypeptide actually this is determined by the type of the side chain. That means the type of the amino acid actually, and the sequence of the amino acids they arrange. Okay, so this the backbone here. They have repeating a C. Here starting N C C N C C N N C C N N C C N. Okay, so the peptide bond is at the NCC. They got one. Okay, that means before between the carboxyl and one amino group. Okay, so it's the peptide bonds here. Okay, so this one is the backbone of polypeptide. Quite important, you must know this is the backbone of polypeptide, because later when we study the secondary structure, you will know the hydrogen bonds that form is between the group CO and the NH group from the backbone of polypeptide. So when like a polypeptide may contain hundreds of amino acids, they join in a specific linear order. So this specific linear order, we call it as the primary structure of protein. So like when you draw it, so this is the N terminus, don't forget, and the C terminus here. Okay, so the amino acids are joined by peptide bond. Okay, and almost infinite variety of proteins molecules is possible. Okay, differing from one another in the number of the amino acids. So the different proteins actually are determined by the number of the amino acid in the polypeptide chain, and also the types. Okay, the types of amino acid. Okay, that used to form the polypeptide, and also the sequences of the amino acid. The sequences of the different type of amino acid that used to synthesize the polypeptide. Okay, so this one will determines the type of the protein. Okay. Okay, so this one is something like so we assume that these 20 amino acids, they are the letters of a protein alphabet. So therefore, in this polypeptide here, okay, so the sequence of the different amino acid, so it's look like a long sentence. Okay, the long sentence. So normally the functional proteins will be the sentence will be meaningful. Okay, so you will study this in the semester 3, the mutation. So how we can the difference between the change of the amino acid sequence. Okay, and then what is the consequence, okay, to the especially the human health. Okay, then now we go to the this part. You have already learned it before. Okay, but we need to study it in detail regarding to the bonding, the form. Okay, protein have 4 levels of organizations. So like the primary structure, okay, secondary structure, tertiary structure, and quaternary structure. Okay, and then for the what we call the tertiary structure and quaternary structure, so they will make the proteins. They will give the proteins a 3-dimensional shape. Okay, the 3D shape. Okay. So there is a close relationship between a protein conformation and its function. For example, a typical enzyme is a globular protein with a unique 3-dimensional shape that allows it to catalyze a specific chemical reactions. So this is here is the active site. If I'm not mistaken, this is an amylase. Okay, amylase. Okay, so as you know, there are 4 main levels of the protein organization, I just mentioned. Okay, all the proteins share the 3 superimposed levels of the structure, known as primary, secondary, and the tertiary structure. So that means all the proteins they will have this tertiary structure. Okay. A 4th level quaternary structure that arises when the proteins consist of 2 or more polypeptide chains. Okay, if the proteins that consist of 2 or more polypeptide chains, so it is in a quaternary structure level. Okay. Primary structure of protein. The sequence of amino acid, okay, the sequence of amino acid that join by the peptide bonds, okay, by peptide bonds, okay, is called the primary structure. So this sequence is specified by the instruction in a gene. The gene in the DNA, okay, they will determine this primary structure of a protein. So as you can see here, here they consist of 29 polypeptide. So the possible number of different polypeptide that can be made from different amino acid is given by the following formula. Okay, P is equals to A power N. So P is the possible number of different polypeptide, and then A is the number of different types of amino acid available. Okay, the number of different types of different amino acids that available to form the polypeptide. Then N is the number of amino acid residues in the polypeptide. Okay, that means how many, that means the polypeptide is made up of how many amino acid residues or monomers. So for the example here, we have only 2 different types of amino acids available, which is the alanine and the phenylalanine. Okay, so from here, P is equals to 2 power 2. Then maybe you don't know which one is referring to A, which one is N. So the 2 types here is 2 A, and then the N here is 2 residues. So therefore, P is equals to 4. So this is the probability or the possible numbers of the different dipeptides can be formed. Okay, and then now we look at the other example. Okay, by using a 3 and 2 different numbers. Okay, so here we have 3 different types of amino acids available used to form a dipeptide. Okay, a dipeptide. So now we want to form the dipeptide. So therefore, P is equals to 3 types. How many types? 3 types. So power dipeptide. Okay, the number of the amino acid residues. So here is to get the answer is 9. So is it 9? So this is the possible number, okay, of the different dipeptide can be formed. Okay, so now you know how to calculate the possible numbers, okay, of the different type based on the different type of amino acids available. Okay, now we go back to just now the example here. This polypeptide they have 29 amino acid residues. So does that mean let's say the amino acids available is 20 types, 20 different types. So therefore, the possible number of the different polypeptide can be formed by using 20 different types of amino acid. okay, in a polypeptide which has 29 amino acid residues. So the result will be P is equals to 20 times 20 power 29. Okay, 20 power 29. The number is too large. Okay, until the calculator also cannot show the number. So from here you already know actually there are what we call like just now we already mentioned. Okay, here where is it? Here almost infinite variety of proteins molecules is possible. Okay. Okay, so the overall conformation of a protein is far more complex. Okay, not only they show no such protein, they only show the primary structure only. No. So the conformation of a protein actually is far more complex. So it can be shown as either tertiary structure or quaternary structure. And then they're involving the interactions amongst the various amino acid, okay, that make up the primary structure. Okay, so therefore the higher order of structure, secondary, tertiary, and quaternary, ultimately derive from the specific amino acid sequence from which is the primary structure. Okay, then if there is one amino acid change, if you see the substitution, okay, of just a single amino acid, okay, in a hemoglobin, the polypeptide chain of the hemoglobin can cause a major alteration of the protein function. Okay, they will change the major what we call the protein conformational change, conformation shape or the three-dimensional shape first. Then once the 3D shape of the protein change, they will change the protein function as well. So for example, here is the sickle cell anemia. Okay, so this is the normal shape of the red blood cell. Sure, they have a normal hemoglobin. So when one amino acid in the polypeptide change, okay, they change. Okay, so when they change, this hemoglobin structure, the three-dimensional shape of the hemoglobin also change. So therefore, the red blood cell will have the sickle cell shape. Okay, we have the sickle shape. Okay, we already know in a primary structure, the amino acids they are joined by the peptide bonds. Okay, so in a linear sequence, so that is a primary structure. So now, for this primary structure, they will further the polypeptide will further coiled and folded into a secondary structure. So they form this secondary structure actually by forming the hydrogen bond. Okay, hydrogen bond only. Okay, but these hydrogen bonds they form actually involve the polypeptide backbone. Okay, so how? Now look at the structure. Okay, some region of the polypeptide attribute secondary structure which is highly regular. Okay, like this one is we know it as an alpha helix. Okay, alpha helix, and then this is the beta beta sheet. Okay, so these two secondary structures are derived from the what we call the primary structure, primary structure, and the hydrogen bonds okay, they are involving the backbone of the polypeptide. So like this one, just now we know this one is the backbone, okay, the backbone of the polypeptide. Okay, NCC, 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 so this one also here, so NCC, 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 NCC. Okay, so the hydrogen bonds formed between the NH with the CO of the backbone. Okay, so this is hydrogen bond. Okay, this one hydrogen bond. So same to the alpha helix. Okay, the alpha helix. So this is the polypeptide backbone. Okay, then the hydrogen bonds that form, okay, is between the NH and the CO. Okay. Now this one clearly, because the hydrogens are in the NH group, okay, NH group, the hydrogen is a slightly positive charge, because of it is more electropositive, and And the other one, the CO, the carbonyl group, so is more electronegative. So therefore, when they arrange like this, so the neighboring one, the CO and the NH group together, they occur along the length of the polypeptide chain. So they will interact to form the hydrogen bonds. Okay, they interact to form the hydrogen bonds and produce a regular shapes such as alpha helix and the beta bitter sheet. Although the hydrogen bond is weak, okay, but as its occurrence is frequent, that means they have more hydrogen bonds they form, so the total effect contributes towards the molecular stability. And this one, as you know, alpha helix and the beta bitter sheet. So how to form the alpha helix? So it's a region where a polypeptide chain forms a uniform helical coil. Okay, and then this helical structure is determined and maintained by the formation of the hydrogen bonds, okay, between the backbone of the amino acids of polypeptide. So they have the successive turn of the spiral coil. Okay, so how to form? Again, is the hydrogen bonds formed between an oxygen from the carbonyl group, okay, with a partial negative charge, and to the hydrogen from the NH group with a partial positively charge. Okay, so that means this one is partially negative. Here is a partially or with a slightly positive charge. Okay. Keratins, okay, the example, okay. Keratin is a fibrous protein, which is entirely alpha helicals. Okay, keratin, and then here is a many becomes fine one. So this is a one polypeptide chain. So they show an alpha helix structure. Okay, and all of these keratin fiber, okay, keratin fiber, they have many these fibrous proteins, okay, they intertwine together, okay, so to form the keratin of hair, okay, or can be found in the skin, okay, and others. So it is a fibrous protein, so they provide the structural support, okay, of the structural components to organisms. Although the hydrogen bonds, they maintain the helical structure, these bonds can be broken. The hydrogen bonds in the keratins, okay, can be broken and allowing the fibers to stretch under tension. So like the telephone cord here can be stretched. So when the tension is released, so this fiber recoil and hydrogen bonds reform. Okay, so this is the flexibility of the hydrogen bonds in the fibrous proteins. So as this diagram show the keratin hair, okay, so they consist of this alpha helix, okay, they have many what we call this polypeptide chain, okay, in the alpha helix structure. So they join and then they are maintained by the disulfide bridge. Okay, so therefore the keratin actually is a quaternary structure. They show the quaternary structure, but the secondary structure play the important role here, okay, for the shape. Okay, so this is the straight hair, and then this one is a naturally curly hair. This is naturally. Okay, so that means they have more disulfide bridges. Okay, but this one you see the disulfide, so here already up to the quaternary structure because it's more than one polypeptide. Okay, but what I want to show you is the entirely alpha helix here. Okay, for the keratin or the fibrous protein here. Okay. Then the other example is the beta bitter sheet. Okay, so for this beta bitter sheet here, they also form the hydrogen bonds, okay, between the CO and the NH group, okay, in the polypeptide backbone here. So here NCC, NCC, so that means this polypeptide is going this way, okay, going this way, and also going this way. Okay, NCC, so that means here is N terminals, here is a C terminals. Okay, So when they make a U-turn, okay, they turn back, okay, on itself. So because they are parallel, in a parallel form, okay, they must run in parallel. So they will form hydrogen bonds. Okay. So if they can be by the hydrogen bonds that can be formed, or the beta Peter sheet they form from the same polypeptide, or can be different polypeptide. Okay, the important one is the two what we call the two regions of the polypeptide chains, they run in parallel. Okay, so this is what we can see. Same, it's very easier to see. So maybe can be for the same polypeptide or different polypeptide. Okay, so how to form the hydrogen bonds in the beta Peter sheet? So same like in the alpha helix. Okay, so they involve carbonyl group and the NH groups. Okay, so you see here the important, they are arranged in a parallel fashion as I mentioned just now. Okay, in a parallel fashion, either running in the same direction or opposite direction. So later you can see if same direction, you can see the 3D structure is like this. They got the arrow is like this, same. Okay, if opposite direction, it's like this. Okay, for example, like this is in a different direction, opposite direction. One is go here, the other go there, going down here. Okay, and then because the adjacent hydrogen bonding, okay, they form in the parallel form, parallel fraction, so the structure is very stable and rigid compared to just alpha helix. So although the beta Peter sheet is strong and flexible, it is not elastic. Okay, like just now, it is not elastic because the distance between the plate is fixed. So they determine the strong covalent bonds of the polypeptide backbone. So when we compare to the proteins with alpha helix, so this beta Peter sheet is stronger. Okay, fibroin, the protein of silk, is characterized by a beta Peter sheet structure, as are the cause of many globular proteins. Okay, so this fibroin here, you can see they have a lot of beta Peter sheet, so that means they're stronger, much stronger. And it is a common for a single polypeptide chain to include both alpha helix and a beta Peter sheet. For example, the spider's silk. Okay, the spider silk, actually they can see this one is the beta Peter sheet. Okay, so this one is alpha helix. Then you can see the arrow here. This is an arrow this way. So that means this one is they run in a parallel fashion, but different directions. Okay, so since the spider silk, okay, the spider silks, they contain the beta Peter sheet and the alpha helix. Therefore, they have these two characteristics. That means the alpha helix will provide the elasticity to the for the spider silk, and the beta Peter sheet will provide the strength to the spider silk. So that means the spider silk, they are strong and elastic. That's why you can see the Spider-Man, they can swing here and there because of the elasticity and the strength of the spider silk. Okay, so once we finish the secondary structure, so we will move to the tertiary structure of protein. And then this tertiary structure of protein is the overall shape assumed by each individual polypeptide chain. So only one polypeptide is involved here. So this three-dimensional structure or the three-dimensional shape is determined by 5 main factors. Okay, that involve the interactions among R groups or the side chain belonging to the same polypeptide chain. You see, just now the primary structure, they only involve peptide bonds, whereas the secondary structure, they involve hydrogen bonds, okay, from the interaction of the what we call the polypeptide backbone. Okay, for the tertiary structure, so the bondings that form or the interactions that form is involving among the R group or the side chains. Okay, so the bonds that involve, so they are, we can categorize them as the weak interactions, right? And the strong covalent bonds. So for the weak interaction is the hydrogen bonds. Again, the hydrogen bonds involved, but this one is between the side chain of the R group. Okay, not the peptide, the polypeptide backbone. Okay, and the ionic bond. Ionic bond is the weak interactions. Hydrophobic interactions and van der Waals interactions. Then the strong covalent bonds is a disulfide bond or the disulfide bridge. Okay, now look at here. Hydrogen bonds form between R groups, okay, of certain amino acid subunit in a tertiary structure of a protein. So from here you can see hydrogen bonds. Here they show you two. Okay, this one is for secondary structure. Okay, because the hydrogen bonds that forms is due to the what we call is from the polypeptide backbone here. Okay, but this one is from the R group. Okay, this is R group. Okay, this is another R group. So they have the hydrogen bonds. So this is what we want to discuss in the tertiary structure of a protein. Okay, so again, same like in the secondary structure. So it's the carbon, like this carbon group of a side chain. Okay, can form a hydrogen bond with and a hydroxyl group. Okay, of a side chain. So like here, like here. Okay, this one between the side chain. Okay, this one. Okay, then this hydrogen bonds also contributes to the three-dimensional shape of a protein. And okay, ionic bond can occur, okay, between an R group as well. So between a unit of positive charge and one with a unit of negative charge. So the ones amino acid R group of amino acids that have positive charge and negative charge, so they are belonging to the basic and acidic amino acid. So from here, where are you? Okay, this one, ionic bond. Okay, so this is charge, positive charge. So this one has a negative charge. Okay, so actually you cannot use dotted line. Dotted line is for hydrogen bonds. You just draw it closely. Okay, closely. So we'll show this ionic bond. Okay, acidic and basic R group exists in an ionized charge at a certain pH. Okay, like just now we go back to the Zwitter ions. Once the pH change, the charge of the amino acids also change. So therefore, acidic R group are negatively charged and the basic R groups are positively charged. Okay, if let's say the pH change, so these ionic bonds will also change. They either is disrupted or they change. So the three-dimensional shape or the tertiary structure of the polypeptide also change. So that means we can say the proteins, the 3D shape of the proteins is changed and the protein is said to be denatured. Okay, so therefore acidic and the basic R group can be attracted to each other, forming ionic bonds. So the ionic bond is a weak electrostatic interaction. So in an aqueous environment, this bond can be broken by changing the pH of the medium. So now you know why it is important for us to maintain, okay, the pH change, okay, in our blood or tissue fluid. Hydrophobic interactions. Hydrophobic interactions result from the tendency of non-polar molecules. Where is hydrophobic? Okay, here, hydrophobic interaction. So it's due to the non-polar, okay, non-polar R group. So you look at here, all of these, they are non-polar R group. Okay, so because of this non-polar R group, they can be excluded by the surrounding water. So this one, this protein, so they're in the cytoplasm. So cytoplasm, they're aquatics. No, they are aqueous solutions. Okay, aqueous solution. So therefore, the water will exclude all these non-polar side chain. Okay, so they were given this structure. Okay, this shape. So this one is known as hydrophobic interactions. Okay, hydrophobic interaction. Okay, like some R group are non-polar and therefore hydrophobic. So if a polypeptide chain contains a number of these non-polar groups, okay, non-polar groups, and is in an aqueous environment, so the polypeptide chains will tend to fold. Okay, like here they tend to fold, so that the maximum number of the hydrophobic groups are, so that means all here they are hydrophobic groups are, so will come into close contact and exclude water. So the water is outside here. Okay, so the one they polypeptide where they face outside, so this one is the polar group. Okay, the side chains are polar group or charged. Okay, so this is because of the hydrophobic interactions. And this is how the globular protein they fold up into a three-dimensional shape. The tertiary structure actually is due to the hydrophobic interactions. Is one of the main factor here. So the hydrophobic groups they tend to point inwards. That means away the aqueous environment. Okay, so they towards the center of the spherical molecules. And then the hydrophilic groups will face outward the aqueous environment, so making the proteins soluble. Okay, the other one is called the van der Waals interactions. These van der Waals interactions, they occurs in the interior of the globular structure. Okay, they're due to the hydrophobic interaction. Okay, hydrophobic is because due to the exclusion of the water molecule. So when these non-polar molecules, they come close contact, so they will have attraction force, and then at the same time, they will have repulsion force up. Okay, so this attraction and the repulsive force, they form between these non-polar groups, so we call it as the van der Waals interactions. Okay, so another one you can look at here. All of this also is due to the most of the non-polar parts. Okay, so they are from the van der Waals force. Okay. Once the non-polar amino acid side chains they are close together due to the hydrophobic interaction, so van der Waals force, van der Waals interactions helps to hold the non-polar together. This is what I explained to you just now. Okay, so another example like the lipid, previously we studied the lipid fat molecule. So fat molecule, they have long hydrocarbon chain, okay, the fatty acid. So when they come to contact, close together, so they will form these van der Waals interactions. They have this strong, what we call the van der Waals interactions. So to bring especially the unsaturated fat molecules together. So therefore, not unsaturated, the saturated, okay, therefore the saturated fat, they tend to be solid in room temperature. Okay, so this is due to the van der Waals interaction. Okay, so because it's only fat, no water, so there is no hydrophobic interactions here. Okay, disulfide, okay, disulfide bonds, or we call the disulfide bridges, okay, are covalent bonds, okay, strong covalent bonds that link the sulfur atoms of two cysteine subunits belonging to the same chain. Okay, and they also can be different, but here is for the tertiary structure, so it's come from the same polypeptide chain. So for example, like this one. Okay, so here got this is the R group of one cysteine. This is another R group of another cysteine molecule, amino acid molecule. Okay, so when they come contact, so they form the disulfide bridge. Okay, so like I told you here, like this one, disulfide. So this is because the sulfonyl group in each R group, they line up alongside each other, so they becomes oxidized. Okay, they oxidize, so they produce this two hydrogen come out. So they are strong and not easily broken. Okay, so the disulfide bond formed when the sulfonyl group is of two cysteine residue react. Two hydrogens are removed, and two sulfur atoms are covalently linked. Disulfide bonds can be formed between different parts of the same polypeptide chain. Okay, this one's for the tertiary structure, or between different polypeptide chains. This one's for the quaternary structure, such as in the antibodies. Okay, antibodies consist of four polypeptide chains. And then these four polypeptide chains, they also help by this disulfide bridge. Okay, then for the same polypeptide chains, the example is lysozyme. Lysozyme consists of 129 amino acid residues. And then about of the 40% of the polypeptide is alpha helical form. And 12%, okay, this is the alpha helical forms here. Okay, and then 12% is beta beta sheet. Then the other, they are irregular chain, the unlinked one. Okay, and then from here you can see they form the disulfide bond. Okay, in the same polypeptide. Okay, between the 16 amino acid, okay, of the same polypeptide. Okay, the last one is quaternary structure of protein. So as you know, they are composed of two or more polypeptide, chains, okay, the proteins, the functional proteins. Okay, that's why I say functional protein. Okay, so this is called the quaternary structure. And then the same types of interactions that produce secondary and the tertiary structure, they also contribute to the quaternary structure. Okay, so that includes all the 5 main factors there. So this is antibody structure. Okay, it consists of 4 polypeptides. Okay, 4 polypeptides. Okay, and then each of these 4 polypeptides, they're joined by disulfide bonds. So the disulfide bonds, they are the strong bonds to stabilize the molecule. Okay, the antibody molecule. Hemoglobin, this one again, a hemoglobin. You already know, okay, you are quite familiar with this. So it consists of 4 polypeptide chains. So they got 2 alpha and 2 beta polypeptides. Okay, so this one, they have no disulfide bonds that bring the polypeptide chains together. Okay, collagen. Okay, collagen, they also show the quaternary structure. Okay, but they are fibrous types. Okay, the fibrous protein. The hemoglobin and antibodies, they are globular proteins. Okay, the collagen is a fibrous protein. Okay, to show the quaternary structure. They consist of three identical helical polypeptides that intertwine about one another. Okay, by the cross link. Okay, here they have the cross link. Okay, the disulfide between their amino acid. So this giving a long collagen fibrous great strength, okay, to function as the girders for connective tissue in the skin, bone, tendons, ligaments, and other body parts. Okay, so especially like this one, you see tendons and the ligaments. So they want to hold the muscle with the bone and the bone to bone together. So they must be very strong. So therefore, the collagen is a fibrous protein. So you must able to differentiate the collagen with hemoglobin or antibody. But normally we focus on hemoglobin and collagen. Okay, then another example is the transtrentin, okay, which is a globular protein. Okay, these globular proteins, they made up of 4 identical polypeptide chains, and then each of these they compose of 127 amino acid. Okay, here's 1, 2, 7. Okay, here's N terminals, here's a C terminals. So here I show you the primary structure, okay, of the polypeptide. Okay, and then this one they show the 4 polypeptide of the transtrentin protein. Here 1, 2, okay, here 3, this one 4. Okay, so you can see here this one they are beta beta sheet. This one is a alpha helix. Okay. So the transtrentin is a globular protein in blood and the cerebrospinal fluid. Cerebrospinal fluid is the fluid that surround the brain and the spinal cord. Okay, they transport vitamin A and hormone thyroxine throughout the body. So like we go back to the formula just now. If we left to chance, okay, we left to chance, so there would be 20 power 127, okay, different ways of making a polypeptide chain, okay, with the 127 amino acid residues, okay. But okay, the precise primary structure of a protein is determined not by the random linking of the amino acid you simply arrange. No, actually it's by DNA. Okay, for example, here I only show you the part of the DNA. Okay, so this is the nitrogenous base. So from here is like the sentence, okay, the information, the meaningful information. So then later this meaningful information will be transcribed, okay, will be transcribed into the mRNA. Okay, so that means this mRNA, they also they mean they copy the genetic information, okay, and then to be translated, okay, to be translated into a polypeptide or into protein, okay. So this sequence, this genetic information here will determines the sequence of the amino acid in a polypeptide chain, okay. If mutations occur, if they say this G, they change to C, then this one C mRNA will be becomes G. So this codon they will be different. So this amino acid serine so will be different. They will be changed. Okay, let's say maybe they change to phenylalanine. When the primary structure of the proteins change, so they will affect the secondary structure, tertiary structure, and also the quaternary structure if they have the quaternary structure. Okay, then the proteins that form, they either is can be function or they cannot be function. Okay, or totally no proteins is formed. So you will study this in the next subtopic. We will talk about the DNA and the RNA, and you will study this transcription and translation during the semester 3. Okay, denaturation and renaturation of proteins. We already finished this level of 4 level of organization in protein. Then now we want to see how is the protein, the 3-dimensional shape that can be denatured and renatured. Okay. So the biological activities of a protein may be affected by the changes, like just how we mentioned, they change the 3D shape. Then the 3D shape becomes disordered. Okay, like here, they become disordered. So this 3D shape, the functional protein, but now they undergoes denaturation, so they becomes disordered polypeptide. Okay. So such changes in the 3D shape of protein and the accompanies loss of each biological activity is called denaturation. Then the denaturation is occur when a protein is heated, subject to the pH changes, radiation, or we treat with a certain chemical. So this, for example, strong acid, alkaline, heavy metals, organic solvent, and detergent. So all of these factors, they will denature proteins. They will change the 3-dimensional shape of the protein. For example, here during heating, kinetic energy is supplied to the protein, causing its atoms to vibrate violently. Okay, so that disrupting the hydrogen and the ionic bonds, so as well as the disulfide bonds. Okay, so like this is the normal one. Okay, now they become disrupted already. Okay, they're broken down. Then after that, they will form the cross link. Okay, the disulfide bond. Then after that, they becomes insoluble. So the protein is said to be denatured. So coagulation of the proteins then occur. So if this type of denaturation is irreversible. Okay. Ionic bonds are disrupted and the protein is coagulated when treated with strong acid. So when you add the strong acid, so you see the bonding, the ionic, especially the ionic bond. So this is the ionic bond here. Okay, when we treat it with hydrochloric acid, which is a strong acid. So you see, they disturb the ionic bond. Okay, so the structure also change. So the protein is denatured. Okay, when we add heavy metals, okay, the heavy metals is mercury. So they also will form the strong bonds, okay, with the charged group, okay. So they disrupt the ionic bonds, okay, and reduce the polarity. So increase its insolubility. See, they form, so precipitate occur, precipitate. So that means the protein will be precipitate in the solution. If we use the organic solvent, for example, ethanol, okay, so they disrupt most of the hydrogen bonding, right? Hydrogen bonding. Okay, you see they disrupt already. So the three-dimensional shape of the protein also change. The protein is denatured. And denaturation generally cannot be reversed as we discussed before. But however, under certain conditions, some proteins have been mildly denatured, okay, have returned to their original shape. They can be renatured. Okay, when the normal environmental condition were restored. So this is known as renaturation. So for example, like here, we heat milk, okay, and then this is the protein they found in milk. So in the low temperature or the moderate temperature, so the proteins, the 3D protein shape undergo denaturation, okay, but we just heat the milk maybe to 50 degrees Celsius like this, then we turn off, okay, we remove the heat. So when the environmental, that means the temperature is restored to the room temperature, so the proteins are able to restore each three-dimensional shape, each normal three-dimensional shape. So renaturations occur. Okay. So when we compare to this one, no, to the 8 just now, the one is permanently denatured. Okay, and the last part for the protein is the how we classify the proteins according to the structure, composition, and function. Okay. So the classification of proteins according to the structure. So we can normally we classify them into two groups, two main groups here. Okay. So one is a fibrous protein and the other one is a globular protein. For the fibrous proteins, they perform the structural function in cells, so such as the collagen, okay, silk, keratin, and myosin. So this protein actually they are insoluble in water, okay, and then physically tough. Long parallel polypeptide chains cross-link and intervals forming the long fibrous. Okay, and then from here, the secondary structure they play the most important role in this formation of fibrous protein. But however, this type of proteins they reach the quaternary structure. So for the globular proteins, the tertiary structure they play the most important role here. Okay, so that the proteins they can be easily folded to form the spherical shape, okay, the three-dimensional shape, and easily soluble in water. So examples are enzyme, antibodies, hemoglobin, myoglobin, okay, and so on, and the hormones. So here actually, some of these proteins they reach quaternary structure, but the tertiary structure they most important to determine the shape of this protein as a globular proteins. For intermediate one, they are fibrous, but they are soluble. For example, the fibrinogen, okay, which is used in the blood clotting. Okay, if you want to classify the protein according to the composition, so they can be classified into the simple protein and the conjugated protein. Simple proteins mean the proteins or the polypeptide only consist of amino acid in their structure. In contrast, the conjugated proteins are the polypeptide that bound with non-protein material. So this non-protein materials is called a prosthetic group. So this type of protein is called conjugated protein. For example, in a simple protein, so you can see the albumin, globulin, okay, all of these are collagen, elastin, keratin, all of these, they are known as simple proteins, okay, because they contain only amino acid in their structure. When we compare to the conjugated protein, you see, very easy, we use the simple one, hemoglobin. Hemoglobin, as you know, they contain a polypeptide chain and the heme group. So the heme group is the prosthetic group of the hemoglobin. Okay, heme group, they contain the iron, porphyrin. Okay, the structure, and also others, what we call this is enzyme, okay, and called dehydrogenase. So they have the prosthetic group, which is the zinc. Okay, so this is permanently, this one is permanently attached to the enzyme. Okay, so this is how this is what is conjugated proteins. Then we also can classify the proteins according to their function. If the proteins they have the catalysis function, so we can classify them as the enzymatic proteins. Okay, if they are, for example, the silk, the one just now, the silk and the collagen, so we can classify them as the structural protein for support. Okay, and then if the ones they found on the plasma membrane, so as the receptor, so we can call it as a receptor proteins. Okay, if the one like the actin myosin for the muscle contraction, okay, for the movement of limb, so we can classify them as contractile and motor proteins. And then the antibody is classified as a defensive proteins, and so on for the rest of the body of the protein. Like hemoglobin, we classify them as the transport protein. Okay, so for the classification depends on which purpose. What do you want to classify them? Either according to the function, or their structure, or their composition. Okay, so in summary, this is what we have learned in this video. I know this video is quite long. Please take time to go through and watch this video carefully. Okay, this is the amino acid structure. Okay, so this is how they form the deuterions, and they can act as the pH buffer in different pH solutions. So this is like different polar, different side chain. Okay, they have a non-polar side chain, which is for the hydrophobic amino acid, and then we have the polar side chain for the hydrophilic acid, hydrophilic amino acid. Okay, and also the electrically charged side chain for the hydrophilic amino acid. Okay, and then they got two. One is the acidic, one is the basic. And also how's the amino acids they can form dipeptide and the polypeptide, okay, through the condensation. Okay, and then how's the formation of the peptide bonds here, and also how's the polypeptide undergoes the hydrolysis to form the amino acid. Okay, so this is the structure of a polypeptide, and then the 4 levels, okay, of organizations, and then the bondings that involve in the tertiary and the quaternary structure. Okay, and then the denaturation of proteins, and then some proteins can be renatured, okay, into the normal protein, and also the classifications of the proteins, okay, based on the structure, composition, or functions. So this is the checklist, okay, found in your learning outcome, so you can check, okay, on your for your own understanding. And the next lessons will be nucleic acid. So that's all for this video, and thank you very much. Bye.